Hello dear learners, welcome to all of you. So in our previous lecture we have seen MBRT test. This test is basically considered as a dye reduction test. In a similar way, today we are going to see new test, that is Resazurin reduction test. It is this principle of this test is somewhat similar like MBRT test, but in this particular test the time of incubation is we have to keep the time of incubation at constant. Okay, so we'll see details regarding Resazurin reduction test. Okay, so this basically this test is known as RRT. Okay, so Resazurin reduction test is another method of dye reduction test, and the principle of this test is nearly similar to methylene blue reduction test. So in MBRT the time for reduction of dye is measured, while in RRT at a fixed period time specific shades of color and its intensity is measured. So as far as principle of this test is concerned, so it is somewhat similar to methylene blue reduction test. In that we have seen that reduction of this dye methylene blue is basically depends upon redox potential and microbial activity. In similar way, in Resazurin reduction test also, the reduction of dye depends upon microbial activity as well as redox potential, but here we analyze the test at fixed period of time, and in this test we have to observe the specific shades of color and its intensity is measured. So there are two variations in RRT of testing milk. So this test has two variations. So one test is 10 minutes RRT test that can be used as a rapid platform test for quick assessment of milk at raw milk reception dog. So basically this test has two variations. So first is rapid test. First test is considered as rapid because it is performed within 10 minutes. It gets performed within 10 minutes. The ordered one is 1 hour RRT test, and this test is somewhat longer, and so as I previously mentioned that in this particular test we have to fix incubation period or incubation time. Okay, so we'll see what is the basic principle behind this Resazurin reduction test. So unlike methylene blue, the Resazurin undergoes reduction through a series of color changes, usually blue, purple and lavender, pink before completely getting reduced to colorless. So in this particular test, there is a Resazurin dye that this dye undergoes reduction through series of colors or series of shades like blue, purple and lavender, pink before complete getting reduced to colorless. So in case of methylene blue, we had seen that methylene blue dye get directly get decolorized to colorless, but here in case of Resazurin there are different shades of color we can observe after when the time goes and reduction get started. So Resazurin dye, which is blue color at oxidation reduction potential of plus 0.3 volts, undergoes an irreversible change to pink colored compound. That pink colored compound is known as Resazurin. When the redox potential reduces to plus 0.2 volts, and when the redox potential is reduced to further plus 0.1 volt or less, the color of dye changes to colorless, and that colorless compound is known as dihydroresorufin, which is reversible reaction. So in this particular test, this test again is dependent upon redox potential or oxidation reduction potential. So first, initially the redox potential is higher, and as the dye get decolorized or as the incubation period means after several time, so redox potential get reduced to in stepwise manner, and we can see different shades of color as the redox potential get changed. So usually the degree of reduction of this dye is measured after fixed time of incubation of milk sample in presence of dye. So in this particular test, so we have to fix the time of incubation. Okay, so the reduction of dye to a particular shade of color is dependent upon extent of depletion of oxygen by metabolic activity of microbes. Obviously, the reduction of dye is definitely dependent upon oxygen concentration, and the oxygen concentration is again depend upon metabolic activity of microbes. The color change is measured with the help of Lobby bond color comparator and standard Resazurin disc. So you can observe the color change with these two aids, that is Lobby bond color comparator and standard Resazurin disc. So how to prepare standard solution of Resazurin? Okay. So you have to take one tablet of Resazurin and you have to dissolve this tablet in 50 ml of cold sterile glass distilled water by gentle heating to facilitate the dissolving. Okay, and this is bench solution for direct use and should always be used as a fresh. So what you have to do, you have to take one tablet of Resazurin and you have to dissolve that tablet in 50 ml of cold sterile glass distilled water, and you have to provide gentle heat to this particular solution. And what you have to do, you have to prepare this dye freshly. Alternatively, dissolve 0.05 gram of Resazurin powder in 100 ml of distilled water, and you have to boil that solution up to 30 minutes. So this will make a standard solution of 0.05%. We should always keep in cool and dark places, and this solution is stored in an amber colored bottle, and the main solution, that is 0.005% for regular use, should be prepared freshly by diluting the standard solution with distilled water. So how to prepare this dilute solution? For example, if you have to just take 1 ml of standard solution with 10 ml of distilled water. Okay. So in this particular test, you have to first prepare standard solution, and you can use that standard solution whenever it is necessary. Okay. Then procedure. So how to carry this test? So you have to first take 10 ml of milk into test tube, and in that you have to add 1 ml of working solution of Resazurin solution. Okay. So put airtight closure to prevent entry of oxygen. So you have to put some stopper at the mouth of the test tube to prevent entry of oxygen. Then invert the test tube to mix milk and Resazurin solution. So you have to mix this dye with milk and place the test tube in a thermostatically maintained water bath at 37 degrees Celsius, and note down the time of incubation. Okay, so it is kept for 10 minutes for 1 hour, and at the end of incubation, match the color of the milk with one color of standard Resazurin dish. So what you have to do, you have to add this dye to milk, and you have to keep this entire solution at a fixed temperature at water bath, which is maintained at 37 degrees Celsius, and at the end of incubation you have to observe color change, and you have to observe that change color with standard Resazurin disc or some other colored standards. So this is chemical structure of Resazurin, and its scientific name is 7-hydroxy 3H-phenoxazine 3,1,10 oxide. Okay, so this is chemical structure of Resazurin. Again, this is the next slide showing the compound Resazurin, that is the intermediate, and its scientific name is 7-hydroxy 3H-phenoxazine 3,1 sodium salt. So this is basically intermediate compound. So as seen in MBRT test, here we can also grade the milk based on RRT, and these are the some standards that are given by BIS, that is Bureau of Indian Standards. So if the color change is observed in 4 hours and some higher time, then the milk quality is considered as an good. Then next, if the reduction of color change get observed at more than 3 hours, then the milk quality get considered as unfair. And if you get sooner decoloration of this dye, so if that dye get decolorized within half an hour, then the quality of milk is considered as an poor quality of milk. So this is the basically grading of milk based on RRT, that is Resazurin reduction test, and these are the standards that are given by Bureau of Indian Standards. Again, grading of milk by Resazurin test. So these are some color discs or colors, and these colors are corresponding to quality of milk. So in that, first disc number 6, and it shows blue color, and that blue color represents excellent quality of milk. Okay, then lilac, it is considered as a very good quality of milk. Then mauve color, it is considered as a good quality. Then pink mauve color, at pink mauve color, the quality of milk is considered as unfair. Then pink color, so pink color is considered as a bad quality of milk. So pink color is indicator of bad quality. Okay, and blue color is considered as an excellent quality of milk. And if the color of this it gets reduced or it gets changed to white, so that quality of milk is considered as an very bad. Okay, so this is basically grading of milk based on different colors. Okay, so advantages of this dye reduction test. So what are the advantages of this dye reduction test, that is MBRT as well as RRT? So these tests are used for estimating suitability of milk for liquid consumption. Okay, so basically these tests are determine whether the milk is suitable for consumption or not. So these tests are cheaper and less time is required. So these tests are performed within very short time span, and they are not very expensive, they are cheaper. So in case of SPC, clumps of microbes are recorded as a one colony, whereas the rate of decolorizing of dye is due to combined metabolic effect of each bacterium in the clump. So with the help of this test, the activity is measured rather than number of bacteria. So basically in this particular test, we can only access the quality of milk. Unlike artificial media used in standard pet count, milk is the natural environment for microbes is present. So in case of RRT, the results can be measured in very shorter time. So some of the bacteria capable of reducing the dye may not develop colonies or medium used in standard plate count. So as there are advantages, there are some disadvantages for this test. So rate of reduction of dye varies considerably, and it is related to species and the rate at which different microorganisms grow at particular temperature. Okay, so basically this reduction potential of this dye is vary as the microbes, different microbes get changed. So this reduction potential is definitely depends upon which type of microbes they are present in milk. Okay, so inhibitory substances like penicillin and other antibiotics prevent growth of bacteria and thus increases the reduction time. So there are some antibiotics, they reduce growth of microbes, and by reducing the growth of microbes, the reduction potential somewhat slower. So as reduction potential gets somewhat slower, the reduction time get increased. So this test is basically not suitable for classifying milk with low bacterial count or less than 100 to 5 per ml. So basically this test is not suitable for low bacterial count. So reduction capability may be vary because of variation in proportion of bacteria carried into cream layer by the rising fat globules. Okay. Then next drawback is or next disadvantages are this test do not give indication for type of microorganism present. So basically this test only means we cannot check which type of microbes are present in milk sample. So basically this test is qualitative test. The temperature of incubation used during this test is not the optimum for majority of microorganisms present in milk. So this test again not suitable for testing quality of pasteurized milk intended for processing because of low number of microorganisms. And this test require continuous attention until reduction takes place. Okay. So these are drawbacks, and here we have finished Resazurin reduction test. Okay. Thank you.